Okay guys, let's get into most exciting part of video, and that is chemoenzymatic synthesis. Let's try and understand first step. In first step, we take alpha naphthol and epichlorohydrin. Now what happens is we get two products. We get an glycidyl ether and an alcohol. Now let's understand how do we get it. Now this is Cl part. So I forgot to mention that we also have got to use a bit of pyridine because it's a base catalyzed reaction, as we are going to remove HCl from here, right? So HCl is going to go out and that is going to happen owing to pyridine. Alright, and what basically we do is just we take this and stir it for 24 hours at ambient temperature and we get the product. And as I told, the product that we get are two things. One is glycidyl ether and the other one is alcohol. Now let's try and understand how do we get glycidyl ether first. Now in case of glycidyl ether, what happens is this Cl and this H has to go in form of HCl. So let's try it out. So this is H out and this is Cl out and this is HCl out. Alright, and this combines over here. So this would combine over here and would give us glycidyl ether. So we got one of the product. Now how do we get the other product? Alright, now let's try and understand that. Now this is glycidyl ether, right? This is glycidyl ether that we got, and as I told you, the other one was alcohol. So what happens is this ring opens up, and Cl comes in here, Cl gets in there, and this H over here forms OH. Good. So we got alcohol. Now this also is an ether. The earlier one was that of glycidyl ether, that was epoxide, whereas this that we got is an alcohol, right? So alcohol OH. Now this one which is there is the chiral carbon. Now chiral carbon, why? Because we know you have got CH2Cl over here, you have got OH over here, this is H and this again is a different group. Alright, so we have got 1, 2, 3, 4 different groups attached to this carbon. That makes it chiral carbon. Alright, so what next? We got mixture of ether and alcohol. I'm showing this just with alcohol, not with ether. It's understood. The same reaction would also happen with glycidyl ether. Alright, so what happens here is, once we get this product, obviously what we got is mixture. So the mixture is then treated with acetyl chloride. Alright, acetyl chloride. This is CH3COCl. So with acetyl chloride. Alright, and this reaction is done at lower than 5 degrees Celsius and with a bit of chloroform as solvent. Alright, so chloroform, temperature 0 to 5 degrees Celsius, and it takes around say 1 to 2 hours to form product. Now what's the product? What's the molecular model of product? Let's try and understand. Now see what we have got to do over here is we have got to attach this again. See again, another important thing that I forgot to mention is acetyl chloride has to be freshly prepared. Alright, so let's carry on. So this is H, this is Cl, so let's attach H with Cl. So again HCl generated. Alright, and this would combine with O of OH with O of OH to form COCH3, acetyl derivative of the compound. Alright, acetyl derivative of the compound. So this is our compound. Now this is what has to be treated with enzyme. All right, now what we are interested in it is this carbon. Now this carbon, as I mentioned earlier, is the chiral carbon. Now we have synthesized this molecule not in presence of any chiral catalyst, as a result of which we have got both the optical isomers. All right, let's understand the structure of acetyl derivative of product that we synthesized right now. This is the naphthyloxy part. This is the propanol part, which has been converted into ester, acetylated. This OH has been acylated to form CH3, C double bond O. All right, this has been acetylated. That's what we did. All right, and we have got here CH2Cl. Now we will try. Now what we have, since we have not used chiral catalyst, so as I mentioned, we have generated both the isomers. All right, both the isomers has been synthesized. Now let's try and understand the structure of R and S of this. The carbon that we are going to focus is obviously this, as this is the chiral center. All right, let's understand the chirality of acetyl derivative of our compound. Now we know that this is CO, CH3 attached to O. All right, OAC, O acetyl. All right, now we are supposed to focus on this carbon, as this carbon is chiral. All right, now this carbon attached over here has got Cl. This carbon over here has got O. So obviously this carbon becomes the medium as per atomic weight ranking, whereas this carbon becomes the smallest. All right, why? We know Cl atomic weight wise is much more than oxygen. So now LMS, now this is the largest. Now as it is directly attached to oxygen, so this carbon, this group becomes the largest group. Then comes this group, medium, as it is attached to Cl, and this becomes the smallest group. All right, so LMS goes this way. Now it is all in towards right direction, towards clockwise. So this is R isomer. Now suppose, as I did in my earlier video, let me try to topple it. All right, and then let's see what happens. Now I just topple it. All right, when I topple it, you see that Cl, position of Cl is the same, position of oxygen is the same. Only the thing is O has gone down. All right, so now the large, medium, small is counterclockwise. Now since it is counterclockwise, so it is S isomer. All right, so we have got, now as I mentioned earlier, now for synthesizing acetyl derivative, we did not use any chiral catalyst. Now since we didn't use any chiral catalyst, so we are ending up with both R and S. Enzymatic aspect of this molecule. Now we know that our main interest is this carbon, as that carbon is chiral. Now this has got both 50-50. This has got S isomer and R isomer in the ratio of 50:50. Now let's understand chemoenzymatic process. Now as name suggests, chemoenzymatic process, this is the chemical, and we are also going to use enzyme for this reaction, so we call it as chemoenzymatic process. Now it has got two isomers, two optical isomers, say S and R, and this, assume this to be S isomer. All right, so they are enantiomer of each other. Now we would assume this to be S isomer, and assuming it to be S isomer, we would show the reaction on this. All right, so let's understand. So what are the ingredients that we use for this chemoenzymatic process? We use solvent which is diisopropyl ether. We use a nucleophile which is butanol. This is the nucleophile that I would be using, which is butanol. What is butanol? Butanol CH3 CH2 CH3 CH2 CH2 and then again CH2OH. 1 2 3 4 4 carbon, so but, and since OH, so butanol. Alright, this is what I am going to use for our reaction as nucleophile. Alright, along with that, most important part is enzyme. Enzyme is lipase amino PS. Now this enzyme is highly selective towards S optical isomer. Alright, so and what happens is, what we do, how do we do? We take this, we take this, we take diisopropyl ether and enzyme lipase amino PS, and we stir it at ambient temperature for 9 days. So what happens? This is butanol and this is ester of S isomer. Alright, now since we have used enzyme, so enzyme being selective is only reacting with S isomer of acetate. Alright, so now let's understand how does it happen. Now this, obviously butanol has to come closer, then this bond breaks. No way its mechanism, it is just model. Alright, so this then goes there, combines here to form OH. Alright, and obviously this acetate part would come and combine with butanol to form butyl acetate. Alright, so we got butyl acetate. So this is butanol it was, and this acetyl part got attached to it, so this became butyl acetate. Now this is the byproduct, so I just take it out. Alright, now what we got is S isomer of alcohol, whereas the R isomer of acetate remained as it is. So as a consequence, we have resolved or separated R and S isomer. S isomer is alcohol and R isomer is acetate. Let's understand why the reaction has to be carried on only till 50%. Now what happens is, we have ROAC, S isomer 50 and ROAC, that is ester of both the isomers in the ratio of 50-50. Now we are using N-butanol as our nucleophile, lipase PS as enzyme. Now lipase PS is highly enantioselective selective for S isomer. As a result of that, what happens is, when we go to 50% conversion, the entire 50 of S isomer gets converted to alcohol, whereas the ester remains as it is. Alright, now and the most interesting part is, it's so very enantioselective selective that we get optical purity of greater than 95% EE. EE is enantiomeric excess. Now what does that enantiomeric excess means would be explaining very soon. Alright, now so the results that we get is 50% of as ester which is there gets converted to S isomer, and 50% of ester remains unreacted. Now both the isomers, since lipase amino PS is highly selective towards S isomer, so both the isomers we get of very high optical purity, that is 95%, 95, greater than 95% enantiomeric excess. Alright. Having understood the reason why we should be carrying on these reactions till 50% conversion, next mission is to know the method to find composition and enantiomeric EE percentage. Now, enantiomeric EE percentage, 95% EE. Now, if we say 95% EE, what does it actually mean? Now, it just says that S isomer is 95% excess that of R isomer. But how do we calculate ratios of R is to S? This is the method. You just you do this, divided by 2, what we get is 97.5. Now that is the ratio of S isomer. Alright, now since this being the ratio of S isomer, how do we find ratio of R isomer? From 100, we just subtract 97.5. What we get is 2.5. So the ratio comes to 97.5 is to 2.5. 97.5 S isomer and 2.5 R isomer. Now, EE percentage. Just subtract 2.5 from 97.5. What we get is 95%. So this is called as enantiomeric excess, and this basically is the ratio of two isomers. We have synthesized optically pure R and S isomer of this compound. Alright, but that R and S isomer of the compound that we have synthesized may have some impurity. Now let's take example of S isomer of alcohol that we have synthesized. Alright, 1-chloro, 3,1-naphthyloxy, 2-propanol. Let's take S. Now assume that S isomer of alcohol has some impurity in it, which I have written by small r. Alright, so what we do is we convert it into MTPA ester. MTPA ester. Now what is MTPA? MTPA, let's understand MTPA. M stands for, see, this is CH2COH, acetic acid attached to phenyl group. Alright, so, and this is the alpha position. So we have got alpha methoxy, alpha trifluoromethyl, phenyl acetic acid. Alright, now this obviously is chiral, as we see to this carbon 4 different groups are attached. So this is chiral and this is the enantiomer of that. Alright, being mirror image. Now what we do is we take our compound optically pure which has got traces of impurity, say of R isomer. So we make an ester, we synthesize an ester, MTPA ester of this molecule. Now when we synthesize this molecule, so obviously S would combine with S of this to form SS ester, S of this and S of this would form SS, and this impurity which is there, that also would combine with this to form SR. Alright, so it had formed diastereomer. Now it has two chiral center, one of this and one of this. Now once if you make a diastereomer of a chiral molecule, it becomes little easy for separation on HPLC column. So we use HPLC, then we inject the diastereomer that we have synthesized into HPLC column, obviously chiral column. Now this chiral column can differentiate both the diastereomers. As a result of that, we get two separate peak for both the diastereomer. Alright, so this obviously I pointed, this small r is again of our alcohol, big S is of MTPA ester, and here also both are SS. This S belongs to our alcohol and this S belongs to this. So both the diastereomer separates out very nicely, giving two different retention times. Alright, so this is one, this is one. This is just I've just in order to explain, I have made this. So we normally get peak like this only. Alright, so as a result of that, this would directly give us the percentage of both, and once we get percentage of both, we can obviously calculate enantiomeric excess. In case of propanolol, matter was much easier. Why? Because we already had specific rotation of chiral propanolol in literature. So E for chiral propanolol was determined by comparing observed rotation with that of literature reported value. Alright, so in either case, we found that our enantiomeric excess was greater than 95%. Now there's vast difference in polarity between the two. So S is an alcohol, is more polar, whereas R is an acetate and less polar, owing to which they can be separated by column chromatography, as column chromatography purely depends on polarity. So this being more polar would be retained in column for a longer while, whereas the R isomer or the acetate being less polar would be eluted out readily. Alright, so acetate is eluted out first by column chromatography and alcohol is eluted out later. This is the setup that we use normally for enzymatic reaction where there is no gas evolved during reaction. Alright, and so we have a reactor, a magnetic needle, and if the reaction requires low temperature, obviously it requires low temperature, so we have a water bath, and this is the heater cum stirrer where we can stir this stirrer for the reaction to be uniform, and we also have a heater in case we have got to heat to say 50, 55, 60 degree or so, because we know that enzymes are not stable at temperature above that. Alright, so that's one setup. This is yet another setup. In this case we use bend tube if we expect some gas to be evolved. Alright, this is the setup we use when the reaction mixture is sensitive to moisture. Now we put in calcium chloride guard. Here we put in calcium chloride which absorbs any moisture entering in, and 2 cotton plugs just for protection so that calcium chloride doesn't drop in to reaction mixture or it doesn't drop out. Alright. Once the reaction is set up, then what we do is we monitor the reaction. How do we monitor the reaction? We monitor reaction with the help of TLC plate. We spot the reaction mixture intermittently. We keep on spotting reaction mixture on TLC plate. So how do we do it? Now this is TLC plate. Now it's a glass plate which has got uniformly smeared silica on it. Now this once spotted, then it is put into TLC jar. Now in TLC jar there's solvent over here and there's a filter paper. You see behind this there's a filter paper here. Now the filter paper is put in beforehand so that it absorbs the solvent via capillary action. So what happens is when I put in this TLC plate, there is a positive pressure on TLC plate which forces the solvent that we have taken to move up. Alright, and then it moves up till a particular part which is called as solvent front. Alright, so that I would show you in next video. Alright, so we spotted, the care has to be taken that the spot that we spot is above the solvent that we have taken. Now this silica on the plate is stationary phase and the solvent which is moving is mobile phase. Alright, so jar, filter paper as I told, TLC plate, it has to be covered, otherwise the solvent normally that we use is chloroform or dichloromethane, which are low boiling solvents. Alright, so we have to have lid on it. TLC plate is taken out of TLC chamber, dried, and then put into iodine chamber. Now once put into iodine chamber, the job of iodine is to colorize this spot. Let's understand retention factor. Now retention means retention of material on silica. Alright, now this is a silica bed. Now suppose the compound that we had spotted here moved till here. Alright, and the solvent front moved till here. Now assume that this distance is 1 centimeter and this distance when measured is found to be 0.6 centimeter. So in order to find retention factor, we have got to divide this distance by this distance. So our retention factor becomes 0.6 divided by 1, that is 0.6. Alright, 0.6 is the retention factor. Let's try and understand RF in broader sense. Now what happens is we spotted over here mixture of tri, di and monoester. Now when we spot and move it on TLC, we find that triester moves to the top, diester here and monoester least. Triester being least polar moves this much, monoester being most polar amongst the three moves still here, and diester in between. Now if this distance from here where we spotted to solvent front suppose is 5 centimeter, this is 4 centimeter, this to this is 3 centimeter, this to this is 1 centimeter. Now let's understand the RF. RF, as mentioned earlier, it would be, for this it would be this distance, that is 4.5 divided by 5, so it would be 0.9, and identically for diester it would be 0.6, and for monoester it would be 0.2. Alright, so what we find? We find that the least polar one which was there had the highest value of RF factor, whereas the most polar had least value for RF factor. And if polarity based, as I mentioned, it is likes like, so polar material is being liked by silica, owing to which it is retained over here, whereas this is least polar, not liked by polar material silica, so moves up. Alright, so on basis of polarity we can also say that RF is 0.2 greater 0.2. so if it is 0.2, then the polarity is maximum, and if RF value is 0.9, polarity is minimum. So when we spot, in our case, we will get ester and alcohol. Alcohol being more polar compound, so would be retained more, whereas ester being less polar would be retained less, so would move up. So in terms of RF, we would have high value for RF for ester, low value for RF of alcohol. Once we get an indication that our reaction mixture is giving two products, then we set in for analysis. We set in for HPLC, GLC, whatever analysis, just to understand the percentage conversion. Alright, prior to that, definitely we go in for infrared spectroscopy, where we have difference in peak for ester as well as alcohol. Alright, once we get indication from infrared spectroscopy also that we have formed an ester and even alcohol is present, and the TLC shows two spots, and gas liquid chromatography or HPLC also shows two peaks, one for alcohol and another for ester, and ester increasing with time, that gives us a clear-cut indication that presumably the product is forming. So but that's not confirmation. Alright, for confirmation we have got to go in for column chromatography. Before loading the material into column, we have obviously to get rid of enzyme, solvent, that is diisopropyl ether if we have used, butanol, all those things we have got to get rid of. Now that we get rid using this assembly. Now this assembly uses Buchner's funnel. Now Buchner's funnel is just a funnel with a sintered bed, and it has got a rubber band over here. Now so that makes it airtight, because the flask which is there, we are going to connect it to vacuum pump for making filtration efficient. Alright, so this is the Buchner's funnel. Now it has sintered bed over here on which is placed filter paper. Filter paper again depends upon what Wattman filter paper you want to use, coarse or fine. Alright, having selected that, having this assembly done, we pour in our reaction mixture over here. So we have enzyme removed here as residue, and the filtrate has diisopropyl ether, butanol, and our products. Alright, so our job is not yet done. We still have got to get rid of diisopropyl ether and butanol. That we do by distillation. Once we are through with distillation, we are left with alcohol and ester, assumed to be, not yet proved by NMR. Alright, then what do we do? And then we place it on column, on silica bed column over here. Now mind you, when we are placing that adsorbed silica material on this bed, there is no solvent there. Alright, once it has been placed on it, then we gradually pour our solvent, or rather mobile phase, and now we are all set to proceed for column chromatography. Obviously, till this time the nozzle or the stopper is closed. Alright, so what we do is we load our material over here, and then we fill the reservoir with desired solvent. Say if it was dichloromethane, for TLC if we tried dichloromethane, we can fill this up with dichloromethane, and mind you, till now the stopper is closed, and once we are set, stabilized, then we can open the stopper and start collecting fractions. Then as the solvent, rather I would call it as mobile phase. Why we call it as mobile phase? Because this is what is moving, the solvent which is there, that is what is moving, whereas silica stays as it is, so it is called as stationary phase. So what we find is, as the solvent keeps on moving, or rather mobile phase keeps on moving down, along with it even the components keep moving down. Alright, so what we find is, first the non-polar component starts moving down. Alright, it would move down like that. So this is the next stage. We would find that the non-polar component, or rather in our case the acetate or the ester, is keeping on moving down. Okay, and at the same time even the alcohol is moving, but there's a vast difference between alcohol and ester. So what happens is, this gets collected here in tray. We keep on moving the tray so that the eluted materials are collected in test tubes one after the other, and then once this is collected, but we have to keep on monitor it by TLC, thin layer chromatography, that which is the component which is coming. So what happens is, if your column chromatography is done well, we would find that this would come out first, this would be eluted first, and after that we would get this fraction where there is no elution. This is all being monitored by TLC, and then that tray is kept aside. Then we put in next tray, and then we would find in next tray the alcohol would be collected. Alright, so this way, so I have collected in one tray the ester, in another tray I have collected the alcohol. Alright, so this way I have separated the components via column chromatography. Now next mission B is to analyze it, which is most exciting, because till now we are expecting these two to be, this to be ester and this to be alcohol, but confirmation is via instrumental techniques like NMR is the final confirmation that that's our product. Alright, nuclear magnetic resonance. These are array of TLC plates spotted. Now this was the first fraction we didn't get anything. In the center is the reference sample which shows alcohol and ester as per our reaction mixture. Then here we have got the fourth fraction. In fourth fraction we found that the ester had started eluting out. Alright, so even in the 11th fraction we found ester was still eluting. So these all fractions were then mixed together. Then here again we find that 15th, 19th we found that all these fractions which were there, they were having ester. So those esters were mixed and isolated. Then later on we found from 23rd fraction we found that alcohol also started eluting out. So then what happened is, then when we came to 31st fraction we found that it was purely alcohol which was eluted. So these fractions were separated and those fractions were separated, solvent removed and tested for nuclear magnetic resonance. We have, rather I would say, physically resolved both the isomers. All right. So, and then once we have resolved R and S isomer, then we can carry on further for synthesis of propanolol hydrochloride. Once we get this compound, then in final step in chiral form, whether it be acetate or be alcohol, have to condense it with isopropylamine. Now isopropylamine is, see, it has got, if I hold it like this over here, now see it has CH3 CH3, all right, CH3 CH3 CH and NH2. So this is isopropylamine. Now this has to react with this. Now this, when we react it with this, what happens is it combines over here. Again the same story, HCl has to leave. So what I can do is I can just remove this from here and remove this from here. So this again forms HCl, and once it forms HCl, then it just combines over here to this, give us this compound. Now mind you, this can be easily hydrolyzed to form alcohol. This can be easily hydrolyzed, so we got our this would get converted into acetic acid, CH3COOH. Right, this would get converted into acetic acid. All right. At the end of reaction, the thought that comes to us is maybe we have lost 50%. Now, in this case, in case of propanolol, we know that both the isomers were desired. But suppose in case of drugs, pharmaceutical drugs, where in case of chiral pharmaceutical drugs, 50% is desired and 50% is undesired. So when we are moving on to 50% conversion for getting single optical isomer, we get a feeling that, oh God, we lost that other 50%. So is there any way out so that we can make use of even that 50% undesired one? Yeah, there are. Now, like we have studied SN1 mechanism, we have studied SN2 mechanism. In case of SN1 mechanism, what happens is that undesired isomer, the undesired isomer can be racemized. Racemization is what? That suppose if it is 50% of our isomer is undesired and we are left with it. All right. So what we can do is we can racemize it. Okay. And how do we racemize it? We racemize it by either heating or by chemical reaction. That reaction could be via basicity or via acidity. So what happens is that 50% again gets converted into 50-50 of R and S. Alright, so 25% we have saved. Alright, that 25% again we can separate out. Then remaining 25% again we can racemize and then get back. Now suppose if the drug is really high value product, then maybe such process is feasible. Otherwise, yes, it is exhaustive and it is expensive process. Alright, so that is via SN1 mechanism, and there is also something called as SN2 mechanism, which is more preferable. In case of SN2 mechanism, what happens is, now suppose 50% of our isomer is the undesired one. What we do is we apply SN2 mechanism to it. In case of SN2 mechanism, what happens is inversion occurs. So if we start with R isomer, that gets converted into S isomer, the desired one. All right, so we started with R, the undesired optical isomer. On doing inversion, that undesired R isomer gets converted to desired S isomer. So via SN2 mechanism we can do. Now here it is 100%, right? We are not losing. In this case, this is exhaustive, but this is directly converting. In fact, it's very interesting to know that in our biological system also has the capacity of inversion. Now suppose if we have had a racemic pharmaceutical drug, biological system has tendency to convert the undesired isomer to desired isomer. But then that capacity depends upon biological system. It is all again very much depending upon how the biological system is, how efficient the biological system is. And at the same time, it also depends upon molecule, whether the molecule could be converted. All right, so that's not a new process. So in biology, in nature, everything is available, right? All right, so let's coming back to our point, SN2 mechanism, inversion. So how do we do inversion? So I've written over here, unwanted can be converted to desired one. All right, so how do we do it? We do it via varieties of method. Like suppose if it's an alcohol, that alcohol we convert it to mesylate or tosylate. Now if we focus over here, we have got paratoluene sulfonic acid, paratoluene sulfonic acid. All right, we can also call it as sulfonate. All right, now sulfonate, when it gets attached to any alcohol, now this can help us for SN2 mechanism for 1 degree and 2 degree alcohol, whereas this tosyl chloride is toluene, and sulfonyl chloride, tosyl chloride can help us only for primary alcohols. All right, then we also have methane sulfonic acid. We call it as this molecule we call it as mesylate. All right, so these are all chemicals that can be used for inversion reaction. So the undesired isomer can be converted to desired isomer. All right, so these are hectic processes, but if the product is really high value product, then such processes can be explored. I was very lucky to get introduced to chiral world. Now in case of propranolol that we synthesized, it's okay. In case of propranolol, we saw that one isomer was beta blocker and the other was contraceptive. Now, not very detrimental to biological self. All right, but then we also saw how one isomer was teratogenic and the other isomer was sedative, prescribed to pregnant ladies, and that was thalidomide. All right, like I had presented in my earlier video. So there are so many such racemic mixtures, 50% R, 50% S racemic mixtures, where one is the desired one and the other is undesired. That undesired could be detrimental or would be staying in body, maybe, you never know, and being detrimental later on. All right, so it's always better to have single optical isomer. So it's really good if more scientists get into this process of asymmetric synthesis, that is synthesizing one optical isomer, the desired one, or kinetic resolution, or separating two isomers via chemical reactions as we did in this case, or have any other process by which we get single isomer. Alright, so it is very much essential for us to work towards getting single optical isomer of optical purity. Hope you all enjoyed the video. I'll be keeping on posting my experiences in scientific world and would really appreciate if you also put in your views in comment section. Thank you.